guys, let's do our last chapter on Mendelian inheritance. The objective for this is to apply the basic principles of inheritance and probability to the solution of genetic problems. Now, we're going to focus on Mendelian inheritance. It refers to Gregor Mendel. He was floating around working at the end of the 1800s. Now, when he showed up on the scene, people had a decent idea of what heredity was. It's the tendency for traits to be passed from parent to offspring. And heritable features, we call them, well, then we called them characters. I think these days we're more likely to call them traits. And there are alternative forms of each trait. Now, when Mendel was working, the theories of inheritance were based on something called blending, where parents of contrasting appearance produce offspring of intermediate appearance. And it sounds good at first glance because we all kind of look like mom, kind of look like dad. We're somewhere in the middle. But it doesn't take into consideration, sometimes we look just like mom or just like dad. Now, at the time, they were like, well, that's because, the genetic material is unstable. Now, Mendel just really wasn't a fan of blending. He actually formulated what he called the particulate theory of inheritance, which involves reshuffling genes from generation to generation. He called them particles, he called them factors. We know now these are genes. Now, at the time, genes as something that we were aware of didn't exist, and he didn't have a microscope, but he's absolutely right. He figured out heredity before we knew anything of DNA and chromosomes because he did quantitative science. He counted, he measured. He literally solved how heredity works by counting pea plants. He was on the cutting edge of quantitative science. Before that, people would just, say, oh, well, some of the plants were purple, some of the plants were white, but wouldn't actually say how many of each they had. So here's Mendel himself. Interesting character. He was an Austrian monk and eventually became abbot, so he does all right for himself. But he's a science guy. So he joins a science society, and part of the terms of the society is he had to research something. So he decides to duplicate research that's been done before, only this time he's going to quantitate it. So he's looking at heredity of the pea plant, and he publishes his findings. He presents it to his science society, and they're like, whoop-de-doo, you grew pea plants. And so his work was ignored until after his death in the early 1900s. It was rediscovered and it blew up. Everybody that read it in the early 1900s were like, this is the most amazing thing ever. So Mendel never lived to encounter this, but he is considered the father of genetics. So this is what he was trying to do, okay? For 200 years, people have been crossing pea plants, and they all got the same results Mendel did. And this one guy, T.A. Knight, crossed purple flowers with white flowers, and he noticed all the offspring had purple flowers. But if you let two of these plants cross, he said half have purple flowers, half have white. That purple flowers have a quote-unquote stronger tendency to appear than white. But he didn't quantify his results. And without quantitation, your results don't actually say anything. You need to be able to analyze it. That's where math comes in handy. So Mendel repeated Knight's experiments, and he lucked out actually because garden peas are awesome for studying heredity. There are a lot of varieties available. In fact, Mendel dealt with 22 different varieties that have really easily distinguishable traits that can be quantified. Okay, easily distinguishable. Objectively, determined. You know, it's not a case of the flowers are lilac versus lavender. They're purple or they're white. Peas are also small, so they don't take up a lot of space. They're easy to grow, and they make a lot of offspring fast. Also, they self-fertilize. Let me show you that. Okay, so the bigger petal just kind of sticks straight up. The smaller petal, notice how it curves around the reproductive organs. Okay, so they self-fertilize. You know, they wind up just fertilizing themselves to make seeds. So you wind up with true breeding varieties that are consistent generation after generation. Okay. However, their reproductive organs can also be very easily manipulated, so you can control who's pollinating who. Okay, the way Mendel did this is by cutting the male parts off and sticking them in an envelope, and then hand pollinating flowers with a paintbrush covered in pollen, so that he could pollinate a flower with that flower's own pollen, or he could pollinate with pollen from a different flower. So that worked out very nicely for him. Now, he had a very specific experimental design. The first thing he did was he let the plant self-fertilize for quite some time. And by doing that, he established that the plants were true breeding. Each variety is going to contain only one type of trait. And he named these the P generation. Okay, so at the bottom here we've got a schematic of his experiment. Now we know now that each individual has 2 genes for every trait. So right there we're looking at height. And we know that the tall plant only carries the allele for tall. So its gamete only carries the tall allele. And the short plant only carries the short allele. So the gamete can only carry the short allele. So he lets his P generation, cross. And he does actually reciprocal crosses, where he puts tall pollen on a short plant and he puts short pollen on a tall plant to determine if the parental contribution is determined by who's the dad versus who's the mom. It actually makes no difference in these pea plants. Okay, so he crossed these 2 varieties, and the resulting offspring, he named them the F1 generation. That stands for first filial generation. Okay, filial means son. Okay, and what he found was that all of the offspring were tall. Okay, he let these offspring self-fertilize, and what he got was the F2 generation, second filial generation. Okay, he got 3 tall plants for every short plant. Okay, so for each pair of plants of contrasting varieties that he crossed, one of these traits would absolutely disappear in the F1 generation and then come back in the F2 generation. You know, he called the trait that he saw in the F1 the dominant trait. The one that disappeared and then came back, he called the recessive trait. Okay, and he crossed a whole bunch of different varieties, comparing plants that were alike except for one trait. It was very specific. They had to be different in only one trait. And these are his actual data points. And if you actually do the math, you'll notice it's not a perfect 3-to-1 ratio. It's always like 3.0125 or 2.93669 to 1. But the larger a sample size you have, the closer you get to the perfect 3-to-1 ratio. Okay, so he found that the proportions were consistent. 3/4 of his F2 individuals will express the dominant trait, while 1/4 will express the recessive trait. Okay, Okay, he will always have a 3-to-1 ratio. Now, he's a smart guy, so he knows that the recessive trait doesn't disappear. Okay, it just hides somehow. So he allows the F2 generation to self-fertilize, and he has a look-see at the F3 generation. And let me actually describe this while looking at the picture. So the plants expressing the recessive trait, when they self-fertilized, all he got were plants expressing the recessive trait. So those were true breeding. The 3/4, the 3 expressing the dominant trait, one of them is also true breeding. When self-fertilizing, it gives only the dominant trait. The remaining 2, when they self-fertilize, their offspring have that same 3-to-1 ratio, 3 dominant to every recessive. Okay, so Mendel figures out that 3-to-1 ratio is actually a disguised 1-to-2-to-1 ratio. Let me back up so we can discuss that. Okay, because our offspring, our F1 generation, are all carrying one tall gene and one short gene, or to kind of apply it to other traits, one dominant and one recessive together. Their gametes have a 50-50 shot of carrying the dominant or the recessive. So what you wind up having is, for every 4 offspring, you have one that is carrying a tall gene from both parents. This is homozygous dominant. 2 that are heterozygous, carrying a tall from one parent and a short from the other. And 1 that is homozygous recessive. So you get your 1-to-2-to-1 ratio. Let's back up to where we were. Okay. So that's when Mendel kind of figured out genotype versus phenotype. Genotype refers to the actual genes that you're carrying. So big T, little t. Or you can describe it as homozygous dominant, heterozygous, homozygous recessive. Okay, so genotype refers to genes. Phenotype is the physical appearance, the physical manifestation of the trait. So tall, short, purple, white. What effect does that gene have? Okay, when we look at genotype and we look at it with the T's, the letter symbol chosen for a given trait is the first letter of the dominant version. So here you see big T, that's big T for tall. And big T means tall. The recessive version, short, that's little t, lowercase t. Okay, purple is dominant over white, so for flower color it would be big P for purple, little p for white. Okay, so where I'm going with this is, for genotype, you see a 1-to-2-to-1 ratio. 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive when you cross 2 heterozygotes. Phenotype, you get the 3-to-1 ratio. 3 tall plants to every short plant. Okay. And from his results, Mendel comes up with the law of segregation. Okay, where he figures out that everyone, he calls them factors. We know now that they're alleles, they're genes. Everyone has 2 genes for every trait, and that they segregate during gamete formation. So each gamete only has 1 gene for every trait. For when the sperm and egg recite, or reunite, I should say, in fertilization, the offspring then has 2 copies for every trait. And dominant versus recessive, what makes something dominant versus recessive is when the dominant and the recessive show up in the same individual, which allele actually gets seen. Okay, a recessive allele is always going to get masked if there's a dominant allele there. So recessive traits, the only way you see them is if somebody has 2 copies of that trait. Okay, so this is kind of a more modern description of what Mendel figured out. Okay, every trait in a pea plant, granted every trait in any diploid individual, is controlled by 2 alleles. The dominant allele is going to mask the expression of the recessive allele when they show up together. And alleles occur on homologous chromosomes at a particular spot. And if they carry identical information, they're homozygous. If they carry different information, they're heterozygous. Okay, again, just to remind you about homologs versus sister chromatids. Sister chromatids are identical. Homologous chromosomes carry information about the same genes, or I should say, carry genes for the same traits, but not necessarily the same information. Okay, so when you look at these, one homolog has big G, the other one has little g. Different varieties. Big R versus little r, big S versus little s. This also can show you, this is the dad's chromosome is purple, mom's chromosome is green in a particular individual. And you can see that for gene Y, this individual is homozygous, identical information. For gene P, heterozygous. Okay, so it's not a question of the entire chromosome is going to be alike. It's gene by gene. So Mendel then gets curious about how various traits get inherited together. So he does the same experiment. The previous experiment I told you about, that was a monohybrid cross, meaning looking at one trait, mono. He looks at dihybrid crosses and pretty much runs the same experiment, except this time the plants are different in 2 traits. Okay, and before I show you his results, from the results that he got from this, he came up with the law of independent assortment. This is independent assortment like we saw in metaphase 1 of meiosis, where the homologs line up side by side on the metaphase plate, and they line up completely independently of all the other sets of homologs. Each set of homolog lines up independently of all the others. Okay, Mendel figured this out. Okay, now, what he did was look at pointer options. What he did was look at how 2 traits get passed down together. So he's got, we'll look over here and then we'll go over to the Punnett square. Okay, so he starts with one individual that has round yellow peas. So we've got 2 chromosomes that carry the round gene and 2 chromosomes that carry the yellow gene. So our gametes can be nothing but round and yellow. Our other individual has both recessive traits, wrinkled and green, okay, and homozygous for both. Okay, so our gametes can be nothing but wrinkled and green. Now, our offspring is going to be round and yellow because the wrinkled and green are recessive. So here's our round yellow. Okay, this individual, when it goes through meiosis, well, because of independent assortment, we could wind up with round and yellow winding up together, because the round and wrinkled separate and the yellow and green separate. So I wind up with every gamete. Okay, this is key about dihybrid crosses. Every gamete winds up with a gene for both traits you're looking at. So here we're looking at seed shape and seed texture. Or I'm sorry, seed shape / texture and seed color. Sorry, my bad. Okay, so round and yellow, wrinkled and green, and every variation in the middle. So round and green, wrinkled and yellow. Okay, so we represent every variation of gamete. Okay, so round and yellow, round and green, wrinkled and yellow, wrinkled and green. And what we get is a 9 to 3 to 3 to 1 ratio. Okay, our 9 round yellow ones include this one that's homozygous dominant for both traits, homozygous dominant for seed shape, but heterozygous for seed color. That's this one as well. Homozygous for seed color, heterozygous for seed shape. That's this one and this one. And then heterozygous for both traits. That's this one, this one, this one, and this one. Okay, so I'm going to change colors. Okay, we also have 3 round green ones. That includes, okay, these are all going to be little y, because the green, that's recessive. But we've got the one that's homozygous dominant for seed shape, and 2 that are homozygous or heterozygous for seed shape. Okay, another color change, bear with me. How about green? Green's nice. Okay, we got 3. They're going to be wrinkled and yellow. Okay, this is recessive. So we're going to be homozygous for that. But homozygous dominant for seed color, that's this guy. And then heterozygous for seed color, that's this guy. And this guy. And then there's one that's going to be recessive for both, and that's that one right there. So when crossing 2 heterozygotes, Mendel noticed you always get that same 9 to 3 to 3 to 1 ratio. And Mendel's smart. He can kind of connect the dots and put all the info together. He can kind of put all the info together and figure out what's going on. So he does. And this is right out of chapter 10. I mean, we saw this. He figures out that each set of homologs arranges itself independently of all the other sets of homologs. Oh, and just for giggles and headaches, this is a tri-hybrid cross. Just so you can see that other, people do kind of go beyond how 2 traits are inherited together. We are not touching the tri-hybrid cross. Okay, so let's back up a sec. We've been looking at Punnett squares, but what are they? It's a table. It lists all possible genotypes resulting from a given cross. So, what a given dad and a given mom will contribute to their offspring. So what we do is line up all the possible sperm genotypes on one side and then all the possible egg genotypes. It says lined up on the other side, but more accurately it would be lined up across the top. And every possible zygote genotype goes in the squares. So here's a Punnett square. And it allows us to really easily calculate probability of genotypes and phenotypes among the offspring. Okay. Now, Punnett square, if we show a cross between two heterozygotes because it makes it really easy to see what's going on. But you can do it with a couple of homozygotes too. It just depends on what each parent is contributing. Okay, but Punnett square shows that Punnett square shows that it's a 50-50 chance that you get one gene for a given trait from each parent. 50-50, you get one or the other. So in the case of heterozygotes, you can really see how that works because, with heterozygote, the genes are different. There's a dominant and a recessive. So the chance of getting, in this case, big P versus little p is 50-50. Okay, now as far as what the offspring will inherit, well, that's the chance that you get one allele from mom and one allele from dad. So that gives us the product rule. Write that down, product rule. That's where you calculate the chance of getting two particular alleles together. So you multiply the chance of getting each allele by itself. So 50-50 times 50-50 equals 1 in 25%, or 1 in 4. Okay, and you can kind of see how, I'm going to pull out my pen again, how this works. This particular allele shows up here and here. So you just take that allele and go to all the offspring all the way down. And same thing here. So this one shows up here and here. Okay. So goes down. Okay, and then we'll put this one in blocks. Okay, so oh, I'm circling this one just so that you can see where these letters are coming from, which parent is contributing them. Okay, so put that triangle on that one, and you can see, how this works. Okay, now something I want to kind of note is this one and this one, and that'd be these right here. Now this brings us to the sum rule. I want you to know that one too, the sum rule. The sum rule states that when the same thing can happen two different ways, basically getting a big P from one parent and a little p from the other, you just add the chances of each of these happening. You just add them together. So a quarter plus a quarter equals a half. So a 25% chance of homozygous dominant, 25% chance homozygous recessive, and a 50% chance of heterozygous. And that would be our 1 to 2 to 1 ratio. Now what's kind of cool is by looking at Punnett squares, we can also figure out what's going on. Kind of make heads or tails out of what's going on if we have to do a test cross. What a test cross is, well, if an individual has the recessive phenotype, you know what their genotype is because they have nothing to mask those recessive genes. However, individuals with the dominant phenotype, well, you don't know what they have going on genotypically. They might be homozygous dominant, they might be heterozygous. So what you can do is something called a test cross, where you take a mystery individual and cross it with a homozygous recessive individual. So the way we do this is if your mystery individual, that's this individual right here, if they are homozygous dominant, well, then we know that all the offspring are going to be heterozygous, in which case they are all going to display the dominant phenotype, and that'd be purple. If our mystery individual is heterozygous, I can't turn off the pen, sorry. If our mystery individual is heterozygous, then half of our offspring are going to be purple and half are going to be white. Okay, we can do this with a dihybrid cross as well. I shrunk this down because honestly, for a mystery individual, can contribute one type of gamete. So instead of having 3 other columns that look exactly like this one, I shrunk it down. Okay, if our mystery individual is heterozygous for both traits, you're going to have a 1-to-1 to 1-to-1 ratio. Okay, let me explain. You're going to have a quarter of your offspring are going to display both dominant phenotypes. A quarter are going to display the first phenotype recessive, the second phenotype dominant. A quarter are going to express the first phenotype dominant, the second phenotype recessive, and the last one's going to express everything recessive. If your mystery individual is homozygous dominant for both traits, then all you're going to have is the dominant phenotype. Okay, if your mystery individual, and this covers both of these, but if your mystery individual is dominant for homozygous dominant for one trait, AA up here and BB down here, and heterozygous for the other trait, BB up here, AA down here. Well, whatever trait your mystery individual is homozygous dominant for, all your offspring are going to express the dominant version of that particular trait. Okay. No, thank you. If your mystery individual, okay, whatever your mystery individual is homozygous dominant for, the offspring are going to display the dominant trait. However, the trait that your mystery individual is heterozygous for, you're going to see a 50-50 split, where half of your offspring are going to display the dominant phenotype and the other half are going to display the recessive. Same thing down here, half dominant A, half recessive A. So it actually does have some use, these test crosses. So here's another rundown of Mendel's laws, and I'm just going to literally read this, so you can pause it and read it. Move on when you're ready. Okay. Now, as far as why we all care, well, what happens when things go wonky? Okay, so genetic disorders. These are medical conditions caused by alleles inherited from your parents. Okay, so you got the correct genes. They're just doing funky things to you. So we're going to start with the autosomal disorders. These are disorders from genes on your non-sex chromosomes. So any of the numbered chromosomes. That's chromosome number 1 to chromosome number 22. Okay, remaining chromosomes after 1, 2, 3, 4, blah, 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 to 22, then you've got X and Y. Okay, so autosomal genetic disorders. Okay, some genetic disorders are autosomal dominant, meaning if you carry the dominant gene, you have the disorder. So someone who's homozygous dominant has the disorder. Someone who's homozygous recessive, someone who's heterozygous has the disorder. Someone who is homozygous recessive does not have the disorder. That is the only way you're going to escape the disorder. And then there are disorders that are autosomal recessive, where an individual that is homozygous dominant does not have the disorder. An individual who is homozygous recessive does have the disorder. Where this gets interesting is with the heterozygotes, because they don't have the disorder, but they carry the disorder. So if they have children with someone else who either has the disorder or is a carrier, then their children may have the disorder. And when we look at genetic disorders, we usually don't look at a Punnett square. Punnett square is a predictive thing, what might happen from a given reproductive event. Okay, what we generally do is look at a pedigree, which is kind of a genetic family tree. This is a generic pedigree. It doesn't really say much anything. But I wanted to kind of set the stage before we get into details. Okay, circles are female, squares are male, and stuff that's colored in. A solid colored in shape is affected, so carrying the disorder. More often than not, you don't see the half colored in individuals like you see here. Those are carriers, but frequently when we look at a pedigree, we don't know who's a carrier, so you might see just a big question mark on individuals. Okay, so that's a general pedigree. This is a pedigree for an autosomal recessive disorder. Okay, and notice the dad in that first generation. He's got a dominant chromosome, a dominant gene, but we don't know if he's heterozygous, because if he does have that recessive gene, well, it doesn't show up, so we don't know, so we leave him with a question mark. Okay, so with recessive pedigrees, most affected children have unaffected parents. You can see the 2 affected kids in generation 4 have unaffected parents in generation 3. Heterozygotes have an unaffected phenotype. They do not carry the disease, or they do not have the disease. They might carry the disease. Okay. 2 affected parents will always have affected kids, because 2 affected parents, all they can hand off is 2 disease-carrying recessive alleles. And autosomal disorders, autosomal recessive, also autosomal dominant, both males and females are affected with equal frequency. Also, close relatives, like the cousins in generation 3, close relatives who reproduce are more likely to have affected children, because you are more likely to find 2 people carrying the same recessive allele in the same family tree. Okay. Here's an autosomal dominant pedigree. An affected kid will usually have an affected parent, because someone's got to give them that dominant allele. Heterozygotes do have the disorder. 2 unaffected parents will not have an affected kid. 2 affected parents, if they are both heterozygotes, they can have an unaffected kid. Okay? Now, both males and females are affected with equal frequency. I have a few examples of some autosomal disorders. This one is autosomal recessive. It's called methemoglobinemia, and it's caused by the absence of an enzyme diaphorase, which, In your blood, you've got hemoglobin, which carries oxygen. It also makes your blood pink, well, pinker. And it breaks down at a slow rate to methemoglobin. And you've got this enzyme diaphragmase that converts it back to hemoglobin. People who have methemoglobinemia do not have this enzyme, diaphragmase. And what it does is it turns you blue. Could be worse. Now, the painting, it's actually a photograph that was colored after the fact. That is the blue fugates of Troublesome Creek. And that's the family that brought attention to methemoglobinemia. They are, the family tree is down the bottom. You can see a whole lot of inbreeding in there. They lived up in the hills of Kentucky. And, you know, when there's nobody around, you marry your cousins. So a lot of them wound up being blue. And initially I saw this painting and I'm like, well, how blue are they really? Because, I mean, I could take a Crayola crayon and color in my own pictures blue. So I went in search of some photographs, and the top on the right, that's Luna Fugate, and she is the most recent of the Fugates to be very, very blue. Now this is treatable. I love the irony of this. You're blue, so what they do to fix it is to shoot you full of methylene blue, which is a blue biological dye, and it turns you pink. Go figure. Okay. So this one I'm a little able to be somewhat flippant because it's not really, aside from the fact that it makes you blue and that'll mess with your social life, it's not really a debilitating disease. This one's far more debilitating. And this one is cystic fibrosis. It's actually one of the most common autosomal recessive disorders. 1 in 20 Caucasian people is a carrier. And it's caused by a recessive allele on chromosome 7, which causes chloride ions to be defective and not let chloride ions out of the cell. Problem is, the chloride ions don't go through the cells. Water can't get out of the cell because water tags along with the chloride ions. Okay, so water can't get out of the cells, and that causes really thick, not watered down mucus in the lungs and the pancreatic ducts. It also makes their sweat exceedingly salty. This one's nasty. They take digestive enzymes and applesauce before every meal, and they also do breathing treatments, inhaled steroids and inhaled drugs, along with a percussion vest. That vest he's wearing is beating on his chest and back to kind of loosen the mucus. As it is, though, they're still very prone to lung infections, and someone with cystic fibrosis is not expected to live to see 30 years old. So this one's nasty. Autosomal dominant disorders. I've got 3 to talk about. First one, hereditary spherocytosis. Instead of nice, you know, donut-y looking blood cells with the dent in the middle, the blood cells become spherical and they're very fragile and they burst very easily, especially under any kind of osmotic pressure. It also causes, let me make sure I've got my notes right. It also causes issues with your spleen. Make sure I have this right. Ah, yes. Shows up 1 in 5,000 births, and it causes the spleen to be enlarged. They treat this by removing your spleen, and then you feel fine, apparently. Okay. That one's not too bad. Huntington's disease is nasty. It is also autosomal dominant. It's a neurological disorder, so it attacks your nervous system, and it's characterized by progressive degeneration of the brain. As the brain shrinks and disintegrates, you can see in the picture the normal brain on the right and the Huntington's brain on the left, how diminished it is. But as the brain degenerates, you wind up with losing motor control, some muscle spasms, personality disorders, because so much of who you are is up in your brain. And as you lose your brain, you lose who you are. Eventually, these people become more or less comatose, and then they lose control over their heart and their lungs and die. Now, this one's nasty because symptoms don't show up until most often after 30 years old, at which point people who have it have more often than not already had children and have already passed it on. This one is somewhere in the middle. It's not pleasant, but it's also not horrific. This is osteogenesis imperfecta. It's caused by a recessive allele with a non-functional enzyme to create collagen, which is one of the most common proteins in the body. Okay, osteogenesis imperfecta shows up approximately 1 in 5,000 births. There's reduced skin elasticity, weak teeth, heart valve trouble. The sclera, the whites of the eyes, are tinted blue, but the giveaway symptom is really, really bitter, brittle, easily broken bones. The 2 full body x-rays are newborns. As a matter of fact, you can still see the umbilical cord on the left-hand one. And these kids, I got this out of a scientific journal, and the caption said that the 2 pictures had hundreds of fractures. Now, frankly, I can only see a few. But these kids are born broken. The top middle photo is of a 15-month-old. Those zebra stripes indicate weakness of structure. And then the bottom, the pelvic X-ray, that's a baby, and you can see that broken femur. And these kids are born broken. They, wind up with fractures one after the other. It's rough. But the good news is this is diagnosed frequently before birth, if not then at birth or very shortly thereafter. So these kids, they have to be very careful to not break, and they will break, but they do not have a shortened lifespan. It's just a matter of, being aware of how easy they break. Now this is an association study where they try and match up disease-causing alleles with DNA markers that are inherited along with, but not involved with, causing the disease. In this case, we are looking at 3 DNA markers labeled J, K, and L, and with the exception of one person in generation 1 person in generation 6, that's one up from the bottom, everybody who got marker L has Huntington's disease. That's what we're looking at in this particular pedigree. So the idea is these markers, they're faster, easier, and cheaper to test for than the actual disease-causing allele. So, there's hope there. Now, so far we've just talked about the straightforward stuff. Sometimes when you look around, Mendelian inheritance doesn't really seem to fit what you see. Oh, but it does fit. It's just not entirely black and white. So, some traits are controlled by multiple alleles. We've been talking about traits that only have a dominant and a recessive, but there are a lot that have various forms. My example is ABO blood groups, where there are 3 alleles. Everybody gets just 2, but blood type alleles, that's 3. And there are other traits that have dozens. Okay, so our example, there are 3 alleles. I super A, I super B, these both have a particular sugar, oppositely typed sugars on the red blood cells. They unfortunately also have anti-other type of antibody in the fluid of the blood. That's why getting a blood transfusion, the wrong type of blood, will kill you, because your immune system loses its mind and, then attacks and kills the new blood cells. Okay, so the third allele is little I, which has no sugars on the red blood cells, but antibodies against everything in the plasma. The ABO blood type is also an example of codominance, where you'll notice that I super A and I super B are both capitalized. This is because they are both dominant alleles. So if they show up together, they are both fully expressed. Let me explain. You can be heterozygous dominant and have I super A and I super B. This is actually how you get blood type AB. Now, I like to point this out as well by saying that blood type O is the most common blood type, but it's the recessive version of blood. So what this means is, just because it's dominant doesn't mean it's common. Another example of codominance is roan horses, where if you look close at the blue roan and the strawberry roan here, you'll notice that they don't have gray or pink hairs. If you look close, they have black hairs and they have white hairs all intermixed, or in the strawberry roan they had red hair and white hair all intermixed. Both alleles fully expressed. Now, sometimes what you get actually is a middle ground. Incomplete dominance is where the dominant allele does not fully mask the recessive allele when they show up together. Okay, this is our go-to organism. It's called a Japanese 4 o'clock. And you can actually see the genotype when you look at the phenotype. The homozygous dominant is going to be red, the homozygous recessive is going to be white. The heterozygote is going to be pink, and it's a set midpoint between the dominant and the recessive. Okay, so it's one intermediate version. Okay. Now, someplace where you can see this is with familial hypercholesterolemia. This is genetic high cholesterol, nasty high cholesterol. Someone who is homozygous recessive develops fatty deposits in the skin, and they have heart attacks during childhood, like little kids having heart attacks. The heterozygote does a little better. The dominant allele masks it a little bit. They suffer heart attacks in their late teens and through their 20s. Someone who is homozygous dominant, however, has the normal allele, and they do not have the disorder. Okay. But here's the thing. With incomplete dominance, the heterozygote is a distinct midpoint, and it's the recessive allele showing through. The dominant allele is fully expressed, but it doesn't cover up the recessive allele. It's not to be confused with incomplete penetrance, where the dominant allele doesn't always express. And if it does express, you wind up with varying degrees of expression. Okay. Our example, polydactyly. This is extra fingers and toes. Not everyone who gets this allele is going to exhibit the trait. Okay. These are all examples of people who have the allele. You can see top left, no expression at all. Top middle, there's a little bump on the left foot. Top right, a reduced digit. Bottom left, custom-made shoes. Fully expressed, multiple toes. Okay, someplace where you see a lot of polydactyly is in cats. It's actually kind of prized among some people. Okay, but like you see, these are all people who have that dominant allele, but they don't always express, and even if they do express, they don't express to the same extent. Okay, another thing is pleiotropy. It's where a single gene, frequently mutant, but often inherited, a single gene affects two or more distinct and seemingly unrelated traits, like Marfan syndrome. which is actually due to abnormal connective tissue from fibrillin, the fibrillin gene on chromosome 15. It affects the skeleton, it affects the heart and blood vessels, it affects the eyes, it affects the lungs, it affects the skin, it affects everything. Feel free to pause the video and, read this. Another place where we see pleiotropic effects is with the cystic fibrosis gene, where there are various and sundry systems that are affected by this because the chloride channels aren't working. Another example, sickle cell. Sickle cell disease is kind of wild because the change between the hemoglobin gene in sickle cells and normal red blood cells is the change of one amino acid caused by one change in one single base change in the DNA. Now the sickle-shaped red blood cells, they as you can see, they're kind of bulky. They travel more slowly through the blood vessels and they actually clog the blood vessels. They also have a shortened lifespan. They don't do such an awesome job of carrying oxygen. And you get all kinds of symptoms like weakness, poor circulation, impaired mental function, pain, high fever, rheumatism, paralysis, spleen damage, low disease resistance, and kidney and heart failure. It's a mess. And this is all due to clogged blood vessels and reduced oxygen carrying capacity. We're going to take a quick side trip because kind of makes you wonder why some of these disease-causing alleles are allowed to stay in the population. Well, sometimes the environment changes and rare versions of a given gene function better than the norm, and natural selection will favor that rare allele, which will become more common. Okay? The idea is that rare allele can be disease-causing, but being a carrier for it can offer advantage. Heterozygote advantage. Let me give you an example. Sickle cell anemia is rare in most of the world, but it's really common in Central Africa. Now, heterozygotes get enough functional hemoglobin from the normal allele, so they do just fine. Wear out a little fast when they exercise, but they do all right. Okay. But the parasite that causes malaria can't feed on their red blood cells due to the sickle cell allele. So they're highly resistant to malaria. If you look at the maps, you can see that the areas where malaria is endemic and the areas where sickle cell is common, they overlap. Not perfectly, but they overlap. So the idea is if you're homozygous recessive for sickle cell, well, you die from sickle cell. If you are homozygous dominant and have two completely normal hemoglobin genes, well, you don't get sickle cell, but you die from malaria. It's the heterozygotes that survive. Similar situation, we think, in the cystic fibrosis genes common in northern Europeans. We're talking about people with Viking stock. The heterozygotes are highly resistant to cholera and possibly a greater extent to tuberculosis. Okay. So we talked about pleiotropy, where you have one gene that affects a whole bunch of things. Now we're going to talk about the other half of that, polygenic inheritance. One trait governed by multiple genes, each of which has multiple alleles. Each dominant allele has an additive effect on the phenotype. And what you get with these is continuous variation. So you wind up with, you can kind of see the bell curve in the picture. You have, and that refers to height specifically, but you have a few people that are very short, a few people that are very tall, and everything in between. And the most common phenotypes are going to be right in the middle, right around that midpoint. These traits can be affected by the environment, like skin color. You're affected by sun exposure, you get a tan. Height, the better you eat, the taller you're likely to be. And eye color. So, an example, the picture up top there, skin color comes in an entire spectrum of colors. Now, this is a hypothetical example. Let's say we don't really know how many alleles contribute to skin color, but each contributes equally. So hypothetical example, let's say it's controlled by 2 alleles, or let's say it's controlled by 2 genes. That means 4 alleles. Now, if they're all dominant, you're going to be very dark. If they're all recessive, you're going to be very light. Now, if you have 3 dominant alleles and 1 recessive allele. Now notice, it doesn't matter which gene actually has the recessive allele, whether it's A or B. Okay, the important thing is how many dominant versus how many recessive total. So if you have 1 recessive gene, you're going to be a little bit lighter. Not very dark, but just dark. If you have 2 recessive alleles, you're going to be right in the middle, medium brown. 3 recessive alleles, light. All recessive alleles, very light. Okay, there are thoughts that an eye color is controlled by 2 genes. So, you know, our hypothetical example would really apply to eye color. We also think there are some physical disorders that can be controlled by polygenic inheritance and some behavioral traits as well. So they research that. It might be worth y'all looking into it too if you feel like it. So I said that polygenic traits can be influenced by environment. This is an example. It's Himalayan coloring. Those animals like Himalayan bunnies and Siamese cats that have the pale body, the dark extremities. These animals are actually coded for dark hair all over their bodies. But the enzyme that codes for dark hair is only active below 32 degrees Celsius. So in other words, the dark parts of their bodies are the cold parts of their bodies. Now I don't recommend it. I consider it cruel and unusual, but if you were to shave a Siamese cat down and strap a ice pack to its back, that hair will grow in dark. If you were to strap a heat pack to its tail, that hair would grow in light. Now, bottom right, those are newborn Siamese kittens, and you'll notice that since they've been inside mom, and it's nice and warm inside mom, that they're all white. And just in case you don't believe me that I didn't just grab a couple of pictures of white kittens off the internet, bottom left, that's a cranky Siamese mom with her newborns, and you can see they're all white. And they'll darken up, and the dark will start at their far extremities, the very tip of their tails, the very tip of their nose, and work its way in. Okay, the other, they're kind of the opposite effect on Arctic foxes, where the color, the color pigment creating proteins, enzymes, are only active above a certain temperature. So they're colorless in the cold and the snow during the winter, and they go brown during the summer. Something else I want to note, and the textbook doesn't touch on it at all, is epistasis, where one gene, basically you have 2 or more genes, and they both have to be homozygous or heterozygous dominant. They have to be dominant they all have to be dominant for any of them to be expressed. Our example is corn. To produce and deposit purple pigment in corn, the plant must possess at least one dominant copy of 2 different genes. One gene handles pigment production, so making the pigment, that purple color, and the other one handles putting the purple color in the pigments. You need to have both to actually get purple corn. So, I'm actually going to color this. So, here we've got, we make the pigment. I'll write that down, make. make pigment. Okay, but because our B gene is recessive, we don't deposit it. Over here, we deposit it, but we don't make it. And if you don't have both, you're just out of luck. It's no good. Okay, but when you cross these, we wind up heterozygous for both traits, so we get purple. Okay, and here, you need to, we have 9:7 ratio phenotypically, because if you are homozygous recessive for either of the genes, then your purple will not be expressed, because it's no good if you can make the pigment if you can't get it into the kernels. And if you can get it into the kernels, that's all good, but if you're not making it, that's not going to do you any good. Okay. So, last section, X-linked inheritance. In other words, blame your mom for all kinds of stuff. Okay. The term X-linked is used for genes on the X chromosome that have nothing to do with gender. There are a lot of genes on the X chromosome that you need just to survive. X-linked inheritance was discovered in the early 1900s by a group at Columbia University, and they did experiments with fruit flies, which are awesome because they have the same sex chromosome pattern as humans, and they also go from egg to adult in about 10 days, so you can look at them for generations upon generations. So he is trying to duplicate Mendel's work, and what he notices, he's looking at eye color, red-eyed fruit flies versus white-eyed fruit flies, and he's noticing that all his white-eyed fruit flies are male. He's getting that 3-to-1 ratio, but all of his white-eyed fruit flies are males. So he's like, why is that? So he starts doing kind of a side set of research. Okay, and this is, I'll explain it with how we understand it now. Morgan had to figure this out. Okay, so we've got a male fruit fly, okay? The eye color gene is actually on the X chromosome. So male here, our white-eyed male, is carrying a white X and a Y. Okay? His sons get his Y. His daughters get his X. Mom is carrying 2 X's. Females are double X. Okay, both of her kids get a red X from her. Okay, when our F1 generation crosses, okay, the male gives his Y chromosome to his sons and gives his X chromosome, his red X, to his daughters. Okay. The female gives half of her daughters her red X, half of her daughters her white X. Half of her sons get her red X, and half of her sons get her white X. Okay, so we wind up with a white-eyed male and 2 red-eyed females, and 2 red-eyed females and a red-eyed male.
I mean, hopefully it's something we know nothing about.
Okay, so
Oh, yeah, but that's what it is, that would be very embarrassing.
the only way to get a red-eyed female is to take this fly right here
No, the X does not have to be used.
Right, but it's.
with her
Oh, well, the X would be cool, isn't it?
red X
And the 2K.
and her
The 2K.
white X and this guy, our white-eyed male, with his white X and his Y. And what we're going to get, we're going to run a little Punnett square here. What we're going to get is a red-eyed male, a red-eyed male, A
Yeah, but I guess
white-eyed
the-
male?
The white part, it just seemed to go in the end. So to me,
Yeah.
it looked kind of like this feather.
A
Really,
red-eyed female?
it did look feathery close up. It really did.
And a white-eyed female.
Kind of looks.
Okay, so he figured this out, and that's when he clued in that it must be on the chromosome that only the female is carrying. Okay, here is a standard pedigree for an X-linked trait. With X-linked traits, more males are going to be affected than females, because males are hemizygous. That's Hemi. ZY, it's one word, but I'm running out of space. ZYGOUS, hemizygous, meaning they only get one X, and whatever happens to be on that X is going to get expressed. They're going to display whatever happens to be on that X. Females, on the other hand, to express an X-linked recessive trait, they have to have 2 copies. Okay, now most of the time, an affected boy is going to have perfectly normal parents because mom is a carrier. Okay, for a female to have the characteristic, her dad has to have the characteristic,
But when you see the rate, you can, actually I can always. figure
and
out
her mom has
the
to
rate.
at least
Actually
be
play
a
Scrabble
carrier.
Pretty damn good at it. Your win rate will be a little over 50%. Just a little over. Not a lot.
Okay, now, X-linked recessive disorders also frequently skip generations from maternal grandfather to grandson.
That's absolute cheating.
Now,
But
if a woman has the characteristic, well, since her sons get their one and only X from her, and all she has is the affected X to hand down, all of her sons will also have the
rated way higher than you. then
the
you
characteristic.
only gain a little bit by being, you gain a lot by being.
Okay, some examples: color blindness. Okay, the allele for blue sensitive protein is autosomal. Okay, we see in 3 colors. There's blue, red, and green. And there are people who are blue colorblind. That's a completely unrelated disorder that is equally men and women. Now, red-green colorblind, that's X-linked, and you're going to see a lot more colorblind men than colorblind women. So my husband's colorblind, and this right here in the middle, this is what he sees.
You're doing good. You're way above the average.
Actually, just for giggles. Hey, Dan,
What?
come here for a sec.
If you're 21 or 2200, you're cheating. Right.
Yeah, unless you're.
Since I'm on vacation and my husband's here, we're going to have him, we're going to see how badly colorblind he is.
Thank you, honey.
Okay, these top 3. Do you see any difference between these top 3?
The top 2 look the same except for the far right, and the third one, the blue purplish area, looks a little different.
Okay, thank you. Okay. Now, color blindness, you can't be a fighter pilot, but in the grand scheme, it's not going to wreck your life. This one's nasty. Adrenoleukodystrophy, also known as ALD. It's caused by, it's caused by an issue with the peroxisomes. Okay, either a protein does not move the enzyme that breaks down fatty acids into the peroxisome, or it does, or the enzyme's in the peroxisome, but another enzyme does not get the fatty acids into the peroxisome. What this causes is that fatty acids build up in the cells and don't get broken down, and it causes nervous system damage, and also no adrenal gland function. These kids develop normally until they're about age 5, and then they fail to develop further, and it's kind of a degenerative disorder. These kids do not live past age 5. Or, I'm sorry, They are diagnosed about age 5 and they don't live past age 10. Sorry, that was my bad. They have poor coordination, slow loss of hearing, speech, and vision. And if you've ever seen the movie Lorenzo's Oil, I highly recommend it. It's a set of parents, and they were told that their 6-year-old Lorenzo had ALD and that he was going to die, and they rejected that. And through trial and error, they came up with what they called Lorenzo's Oil. It was a mix of olive oil and rapeseed oil, and they fed it to him, and it slowed his disease way down. This picture on the right is Lorenzo, and he actually passed away in 2008. He died of pneumonia the day before his 30th birthday. As you can see, he's got a feeding tube. He's in a wheelchair. You know, they didn't stop his disease, but they slowed it way down. He lived 20 years beyond the most optimistic prognosis. So his parents consider his life a great victory over his disease, and I'm sure he would have too. More recently, as of 2011, March of 2011, 2 kids were cured by gene therapy 2.5 and 3 years previously. So we're looking at the beginning and the end of 2008. Gene therapy, and I use cured in quotation marks because what they did was they injected a healthy gene for these enzymes into these 2 kids. And I say cured in quotation marks because the damage that they had sustained from their disease up to that point was permanent. There's no turning back the clock. But it stopped the disease in its tracks. There has been no further damage. So there is great hope there. Okay, hemophilia, caused by the absence or minimal presence of clotting factor 8 or 9, causes the affected person's blood to either not clot at all or it clots very slowly. This one is somewhat treatable. Oh, by the way, so the downside of this is you can bleed to death from a paper cut. This one's somewhat treatable. They inject clotting factor 8 and 9. So what you see on the right there, that's kind of one of the unsung symptoms of hemophilia, is that there's a lot of internal bleeding around the joints and swelling of fluid around the joints. That person is not doing such a hot job of taking care of themselves, because that should not happen. The other one, the baby with the huge bruise, that is a newborn who is not getting treated yet. But yeah, that's the problem with hemophilia, is like you can die from a bruise because you never stop bleeding under your skin. You can die from a paper cut. So that one's nasty. Little trivia for you. Queen Victoria was a carrier for hemophilia, and her children carried it into just about every royal family in Europe, except for England. England is clean, but everybody else has it. Okay, mainly because if you look closely, you'll see that they're all really inbred, because royalty has to marry royalty. So there's that. Okay, last disorder, muscular dystrophy. It's caused by absence of the muscle protein dystrophin. What happens is calcium leaks into the cells. What we have over here on the right is normal tissue. Over here on the left is tissue from a kid with muscular dystrophy. Okay, so calcium ions leak into the cells, and it promotes the action of an enzyme that dissolves the muscle fibers. Okay, at which point scar tissue forms. The problem with that is scar tissue chokes it cuts off the blood supply and starves the living tissue. So we wind up with all this pale pink stuff. That's all fibrous protein. Or, I'm sorry, not fibrous protein. It's all fibrous tissue. It's scar tissue. Okay. Okay, so you wind up with a lot of muscular issues. Now look how big that kid's calves are.
460 was $20
Okay.
a year, which was still a bit.
That muscle, that
$6 each time you go to the
bulk of those lower legs is mostly fat. It's not strong.
$10 for the rest of your life. Yeah.
Okay. Also, they have a very tight Achilles tendon and weak muscles in the front of the legs, so they tend to walk up on their toes. Gives them an awkward, very clumsy gait. Also, they have really thin, weak thighs, especially that front part, that quad, that big muscle that holds you up, and weak butt muscles. Okay, again, those glutes on the back, they hold you up. Okay, so because these muscles are so weak, they tend to hyperextend their knees to kind of, lock out their legs and hold themselves up. Also, their belly sticks out because they have terrible core muscles, which gives them a sway back. Okay, so now they need to hold their shoulders and arms basically behind them, just to kind of counterbalance. So they have terrible balance. They fall really often, and it's a progressive degeneration of all the muscles. So these boys, they don't live to see age 20. This is a nasty one. However, there is hope. They've isolated the allele that causes this, and they've tried injecting immature muscle cells into the muscle, and they get 30 to 40% dystrophin production, so that's not terrible. They've also tried gene therapy where they inserted the allele, and only about 1% of the cells produced dystrophin. But, therapeutic cloning's promising, being able to put muscle tissue in there and hope that it does stuff. Okay. So, the last thing I want to say is thank you and good night.